I'm Synapse. I'm a member of MHRC and also associate professor at the Department of Rehabilitation Sciences at PolyU. So first of all, let me introduce MHRC. MHRC is a research center aiming to enhance mental health research and clinical practice, psychosocial and cultural impact, and also policy development in promoting mental health. There are four major research focuses of the center, which include, first, the basic and translational neuroscience research on mental health. Second, the neuroimaging studies and innovative cognitive rehabilitation. And third, the behavioral and psychiatric rehabilitation, for example, mind-body intervention for stress management. And fourth is a psychosocial and cultural aspect of mental health. So no health without mental health. Through high-quality, impactful translational research, incorporating the interdisciplinary collaboration with local and international research, MHRC strives to contributing to improving mental health system and tackling global health challenges such as the COVID-19 pandemic and rapid increase in aging population with mental illness. So today, it is a great honor to have Nobel Laureate Professor Thomas Südhof from Stanford University to deliver a talk entitled The Molecular Architecture of Synapse Formation. So without further ado, may I invite Professor Christopher Chow, our Vice President of Research and Innovation, to introduce Professor Südhof. Professor Chow, please.
Thank you very much. First of all, good evening or good afternoon or good morning, ladies and gentlemen, depending where you are. It is indeed my great pleasure to introduce our distinguished speaker today, Professor Thomas Südhof. Professor Südhof was awarded the 2013 Nobel Prize in Physiology or Medicines for his discovery of the key molecular components and mechanisms that form the basics of chemical signaling in neurons. Professor Südhof is currently the Evan Goldstein Professor, a professor of molecular and cellular physiology and of neurosurgery, and by courtesy, a professor of neurology and of psychiatry and behavioral sciences at Stanford University School of Medicine. His neuroscience research describes how neurons communicate with each other as synapses and how this communication is impaired in neuropsychiatric and neurological diseases. Allow me to take this opportunity to give some basic information about neuronal communication. So in our bodies, there are tiny cell-like structures called vesicles, which transport substances and molecules as signals to other cells in the body. For an individual to think, act, or feel, our brain cells have to communicate by exchanging signals at the small gaps between neurons called synapses. In the 1990s, by studying the brain cells of mice, Professor Südhof demonstrated how vesicles are held in place, ready to release signal-bearing molecules at the right moment. For this work, Professor Südhof and his colleagues were awarded the Nobel Prize in Physiology or Medicine and the Albert Lasker Basic Medical Research Award in 2013. Professor Südhof was born in Germany and earned his medical degree in 1982 from the University of Göttingen. In 1983, he moved to the United States to join the University of Texas Southwestern, UT Southwestern in Dallas, where he received his postdoctoral training and started his own laboratory. The year 1986 was the time when Professor Südhof began his independent research career and became an investigator at the Harvard Health Medical Institute until now. After 25 years at UT Southwestern, in 2008, he moved his laboratory to Stanford to embark on a new research direction to investigate how synapses are formed and to follow up on questions about neurotransmitter release. Professor Südhof's wholesome attitude as a scientist and as an individual is truly appreciable and respectable. In the autobiography written for the Nobel Prize, Professor Südhof reflected on the life lessons he had during his 40-year scientific journey. I would also like to share two short excerpts with you. In the text, Professor Südhof mentioned the criticality for a sense of humility throughout the entire career as a scientist. At the arguably rather advanced stage of his career, he said, "I need mentors and teachers more than ever. I need people who know better than I to tell me when I am wrong and to make me aware of my mistakes. A good research tells scientific truth, and the quest for truth is what makes research ethical," he said. "Quantity doesn't matter very much, nor does the place one publishes. As scientists, we have to intellectually dissociate ourselves from fashion and journals and focus on what is the actual content of a study. What the data really say, we can then take pride and pleasure in work that reports an actual advance." So the journey and valuable qualities in the autobiography are very inspiring. You are most welcome to read further. Now, without further ado, ladies and gentlemen, please join me in welcoming Professor Südhof. Thank you.
Hello, everybody. I'm going to try to share my screen. I'm hoping that you can hear me. And that this will work. Sonata, is this working? Can you just say up or down? Up? Okay, good. So thank you very much for the introduction, which was extremely nice. Thank you for actually reading the piece that I wrote more than 10 years ago now for the Nobel website, which I very much still believe in. And it's wonderful to know that somebody actually read it. I think in science, we are continuing to face enormous challenges in terms of truth. What I want to do today, or what I've prepared to do today, is to talk about a scientific project in our lab. So this is a scientific talk. It's not a general talk. And I hope that I judged this rightly. And I hope that for this audience, this is the right kind of talk. If not, please forgive me. But it was my understanding that I would be giving a scientific talk on the work we've been doing. In order to introduce what I'm going to talk to you about, let me just remind you that we as neuroscientists who are trying to understand the brain are facing an enormous challenge, which is posed by the sheer size of the brain. What you see here is an image of the postmortem human brain. There's 10 to the 12 or more neurons, each of which forms thousands of synapses, or most of which, there's a few exceptions, leading to a vast number of synapses that connect all these neurons in the brain to enormous networks. The complexity of the brain greatly exceeds the complexity of the human genome. But we don't understand the human genome. So it's easy to see why it's difficult to understand the brain, because after all, the human genome is, relatively speaking, probably simpler than the brain. So this is our challenge in neuroscience. And there's many different ways of how people approach the understanding of the brain. It's clear that each of us who is trying to do this can only do a little bit. We really have to focus on one particular aspect, one particular question, and try to get further and thereby contribute just a little bit to an overall understanding that covers eventually the brain. And I can tell you, in my view, we are very far from actually an understanding. Moreover, among neuroscientists, there's a great discussion or even disagreement about how best to approach the brain. But there's one concept that neuroscientists share, which is the concept of the neural circuit. And let me illustrate this to you in this very schematic slide here. The concept of the neural circuit is the idea that nerve cells are connected into networks by synapses. And these networks can be small or big. As you can see here, this is a micro network, a microcircuit, where you have just 3 neurons that are connected by synapses shown in the electron micrograph. One red interneuron is inhibitory, 2 blue are excitatory neurons. So what we are trying to understand in neuroscience is how the brain is functioning as vast networks that are formed by neurons that are connected by synapses and are surrounded by glia. And clearly, this challenge needs many, many different approaches. Our lab is focused on synapses, on the structure that you see here in the electron micrographs. And the synapses are the fundamental computational unit of the brain because they are the ones that not only transfer information, signals from one nerve cell to the next, but they also change that information. They process it. They're very fast, they're very precise, and they're extremely plastic. And they differentially process information. Different synapses have different computational properties. Our lab is limited in its scope in trying not only to understand synapses, but actually trying to understand a little bit of synapses, which is trying to understand the molecular rules that determine the formation and specification of synapses. And the fundamental proposition of this work is that synapses are intercellular junctions specialized for transferring and processing information between neurons. And because they're intercellular junctions, whereas 2 cells connect to each other, we hypothesize that synapses are controlled by adhesion molecules that not only make these 2 sites adhere, but are actually signal, that mediate signaling that is bidirectional. Because of the importance of synapses, many adhesion molecules are described. This shows just a schematic view of a few of them that have been studied. I'm not going to go into the magnitude of these interactions, all these different molecules. Let me just state that it's not currently clear which ones of these many molecules are actually important, what they do. Many of them have been studied at a very preliminary way. For most of these molecules, we don't know what they really are for. And so we're trying to systematically study which adhesion molecules make a synapse and how these adhesion molecules work. We're trying to understand how these molecules as signaling devices actually work in establishing and shaping synapses. And today, I want to focus not on all these molecules, but I want to tell you just about one family, arguably the most important family of adhesion molecules here, called neurexins. And I want to focus on them because these molecules are hub molecules that are sort of the focal center of vast number of interactions, a lot of different activities, okay? And that's going to be the topic of my talk today. And so what I'm going to talk about is first, I'm going to introduce you to the neurexins. Want to make sure you'll never forget them. You might forget me, but I hope you'll never forget the neurexins. I will then tell you about one story, one particular function as a way of digging deeper, of communicating to you how we work, how we're trying to understand this, what our approaches are. I will then tell you about a different function, a different story that is completely different, just to give you an idea about the scope, about the breadth of these molecules. And finally, give you a broader picture. And let me just tell you, this is just a focus on one family of proteins. This doesn't explain synapses. There's no claim here of having a final explanation of anything. I'm really going to tell you about science and action, about how we try to understand how the brain circuits are formed by the connections of neurons with each other via adhesion molecules at synapses, and specifically the central role in this of neurexins in this process. Okay, and now I need to find out why this doesn't work. Okay, fine. So let me start. Neurexins are presynaptic adhesion molecules whose domain structure is shown schematically here. They are composed of two principal forms, alpha and beta neurexins. Alpha neurexins are longer forms of beta neurexins, multiple LNS domains. These are the L's, and EGF-like repeats. These are the E's. Beta neurexins just contain an LNS domain, typical type 1 transmembrane proteins that interact transsynaptically, as I already showed you, with a number of postsynaptic ligands. There are 3 genes, each of which has an independent promoter for alpha and beta neurexin isoforms. So there's 6 principal forms. In addition, which I'm not going to discuss, there's also neurexin 1 gamma from its 3rd promoter. Neurexins are localized to synapses. We first showed this many decades ago. What you see here is the confocal images that demonstrate that they co-localize with both presynaptic and postsynaptic markers. Super resolution microscopy carried out in collaboration with Xiaowei Zhuang at the Harvard University demonstrated that within a synaptic junction, as shown here in multiple images, both in hippocampal cultures and hippocampal tissues in vivo, these molecules, the neurexins, are localized in nanodomains, little pieces. And when you zoom in here, you can see this better. On top is in culture, on the bottom is in the tissue sections. And you can see that in these sections, in these synapses, the green neurexins are part of a bigger junction. So there are these nanoclusters, and these nanocluster sizes they occupy about 20% of a synapse, 15 to 20%, depending on whether it's in vivo or in culture. And they contain about 5 or so neurexin molecules based on the antibody saturation. So neurexins are synaptic, and they're in nanoclusters, and they form basically in a subdomain of synaptic junctions. I don't know if you see these funky lines at the bottom. I don't know where they came from. Hopefully you won't see them. I didn't put them there. First time I ever seen this. There's thousands of mutations in neurexins that have been observed in neuropsychiatric diseases, especially in autism and schizophrenia. And these thousands of mutations are associated with a variety of neuropsychiatric disorders. No mutation is specific for a particular disorder. And as you can see here, these mutations are deletions in the neurexin gene, in the neurexin 1 gene primarily, but also in neurexin 2 and 3 genes that occupy quite a bit of DNA because these genes are so huge, and different deletions are associated with different psychiatric disorders without a specific correlation. And this raised the question whether these deletions are actually biologically meaningful or just basically accidental. In other words, do these deletions actually have a biological consequence? And is there a change in the synapses in which neurexins are that one can measure as a function of these deletions? And so we tested this question in human wild-type versus neurexin 1 mutant neurons with 2 approaches. We used both engineered conditional heterozygous neurexin 1 mutations to have total control of genetic background, and we used patient-derived neurons to test this. And what we found when we did this is that the neurexin deletion had no effect on the actual making of a neuron from a stem cell, which we routinely do, but it did decrease as a heterozygous mutation the levels of mRNA and protein, as shown here. And when we measured synaptic function, we found that the heterozygous mutation caused a decrease in synaptic strength that was highly significant, suggesting that a heterozygous mutation could actually suppress the synaptic strength by decreasing the levels of neurexins. Now, that was a quite unexpected finding because it suggested that somewhat surprisingly, deleting or removing just part of the neurexins from a human neuron, from a human synapse, is sufficient to cause a phenotype. And it led us to ask the question whether this is truly something that happens in the patient's brains, in the patient's samples, or whether this is some quirk of engineering a neurexin mutation into a stem cell. And so we tested this using patient-derived neurons. Fundamentally, what we did was get iPS cells that were derived from patients and from control subjects. And then we made 2 pairs, 3 pairs, each of one patient-derived neuron, one control neuron, and we analyzed them by electrophysiology. And we did not only do this in our own lab, but also the iPSCs were sent to a different lab, which made their own iPS, own neurons, and did the same analysis independently to validate the findings, to be as rigorous as possible. And yes, you can see here these 3 human pairs of patient versus control neurons had similar phenotypes. In every case, there's a decrease in synaptic strength with an increase in the coefficient of variation, which is a measure of release probability. There's a change in short-term plasticity that is identical in the 3 pairs, and this was independently validated in a second lab, which made their own neurons from the same pairs and analyzed them separately. So clearly, deleting just one allele of neurexins, as shown here in patient-derived neurons, is sufficient to cause a biological change, to change the synapses, validating the importance, the significance of this mutation. But it obviously doesn't explain the diseases. It doesn't explain why different patients with the same mutation have different clinical manifestations. There's still a long way to go from understanding that this mutation is clinically significant to translating that to an understanding of what happens in the patients, and translation that we are trying to pursue in order to be able to help the patients in future. Finally, neurexins are extensively alternatively spliced at 6 sites, creating thousands of isoforms. And this alternative splicing, as shown schematically here, is important also because it regulates the interactions of neurexins with various ligands. So much for the introduction. I hope you'll never forget the neurexins in your life. This is what neurexins are like. Now, let me move on and tell you about what I really wanted to tell you about, which is one particular story that is actually related to alternative splicing. This specific function of neurexin is a function controlled by alternative splicing at one particular site called SS4. And that site we chose in our initial experiments that were dated, that we started more than 10 years ago, because this is the site of alternative splicing that controls the highest number of ligand interactions. And the results of SS4 were unexpected, as I will tell you, because they address a key question in neuroscience, a question that continues to baffle many scientists, including us, because it addresses only one part of that question. And that question is that how is the number of glutamate receptors in an excitatory synapse regulated? There's different types of glutamate receptors, as you all know, and how are these different types regulated? And this is an important question because it determines the strength of a synapse and the quality of the information processing, the computational properties of a synapse. So to address this question, we generated mice in neurexin 3, in the neurexin 3 gene that contain a mutation. And this mutation is illustrated here. And it specifically changes alternative splicing at splice site 4. With this point mutation, as shown here, renders the exon that is normally spliced in or out in this alternative splicing constitutively in. So it's always in. It's no longer alternatively spliced. And the same exon is then flanked by LOXP sites. So we can take it out with the recombinase. And so this makes it possible for us to control alternative splicing at this site by recombinase. We have it either completely in or completely out. And then we tested the effect of this alternative splicing control on synapses. And what you see here is first observed in cultures was that surprisingly, there was a selective decrease with the SS4 plus variant of neorexin 3 in AMPA receptor-mediated responses, but not in NMDA receptor-mediated responses, not in iPSCs, demonstrating that this particular alternative splicing controlled the recruitment of AMPA receptors such that with SS4 plus, you would have fewer AMPA receptors in cultured neurons with normal NMDA receptors. It's a critical postsynaptic phenotype and raised immediately two questions. First is this something like a culture phenomenon? Can we trust this result? And second, neorexin is supposed to be presynaptic, but this is a postsynaptic phenotype. Is neorexin really presynaptic? Does it really control the AMPA receptors postsynaptic via a transsynaptic action? So to test this, we used an in vivo approach. We manipulated the splice site 4 in vivo using stereotactic injections of AAVs, as illustrated here in the CA1 region. Then 2 weeks later, we cut slices, and we analyzed these slices by slice physiology, by recording from the subiculum. And in this arrangement, the presynaptic neurons that are manipulated in CA1 form synapses on the postsynaptic subiculum neurons, which have not been manipulated. So this is a purely presynaptic manipulation, allowing us to address these two questions that I just posed. And to our relief, I have to say, we replicated the phenotype. We saw a decrease in the SS4 plus, which is here the CA1 control, in the AMPA receptor response, normal responses when we presynaptically deleted the SS4 into SS4 minus, which makes it the same as in wild type. No effect on NMDA receptor-mediated responses. So this raised the next question, because this is really exciting. It tells us that we can manipulate by presynaptic manipulation a postsynaptic glutamate receptor response. Is this true for all neurexins? We have 3 neurexins. They're very similar. They are identically alternatively spliced. Do they do the same thing? Is this remaining AMPA receptor response that you see here, is that due to the other neurexins? Or do other neurexins do something else? So to test this, we made conditional knock-in mice for neurexin 1 and 2 as well, with the same strategy, changing SS4. And when we analyzed them, we obtained a completely different result, very surprisingly. Now, with the neurexin 1, I showed you previously neurexin 3. With the neurexin 1, SS4 plus, SS4 minus, no change in AMPA receptor responses. But now the NMDA receptor responses were changed. SS4 plus had an increased NMDA receptor response that was normalized by deleting presynaptically the SS4 exon from the neurexin 1 gene. So it's a very, very different phenotype. The neurexin 2 manipulations, in turn, had another phenotype. Now, there was no phenotype in either AMPA or NMDA receptor responses, suggesting that the 3 neurexins are intrinsically different, that they perform distinct functions. And to summarize this, the presynaptic SS4 plus alternative splicing neurexin 1 controls postsynaptic NMDA receptors, such that the SS4 plus upregulates NMDA receptors, whereas presynaptic neurexin 3 alternative splicing does the opposite, it downregulates AMPA receptors, not NMDA receptors. Completely different phenotypes, but they always focus on AMPA or NMDA receptors, on glutamate receptors, which obviously are homologous to each other. And this provides a mechanism of a transsynaptic control of receptor responses. How does it work? So when you look at the interactions of presynaptic neurexins with postsynaptic ligands, there's only one ligand that binds to exclusively SS4 plus neurexins, but not to SS4 minus neurexins. These are cerebellins. There's 4 cerebellins, 1, 2, 3, 4. Cerebellin 3 is not expressed in the forebrain. When we looked at what cerebellins are expressed in the subiculum and hippocampus, we found that cerebellin 2 is by far the most abundant. So we generated cerebellin 2 knockout mice and measured their synapse numbers, because cerebellins are supposed to be involved in synapse numbers, could find no change. And then we measured their synaptic transmission. And what we found was the mirror image of the SS4 plus manipulations. We now found that when the cerebellin 2 was deleted, that there was an increase in AMPA receptor EPSCs, as though a presynaptic inhibition by norexin 3 SS4 plus had been ablated, and there was a decrease in NMDA receptors, as though a presynaptic enhancement by norexin 1 SS4 plus had been ablated. So this suggests that indeed the cerebellin 2 is the major transducer of the presynaptic norexin 1 SS4 plus signals. Do these changes in AMPA and NMDA receptors occur in the same or in different synapses? We use minimal stimulation to test this, and they do occur in the same synapses. Happy to discuss this afterwards if there's time. How do the norexin 1,3 cerebellin complexes work? This is now the critical question. How is it possible that 2 different norexins use the same transducer to have 2 different effects? So when you look at the interactions of the cerebellins, you can see that cerebellin 4 binds to DCC or neogenin, but cerebellin 1 and 2 bind to a different receptor called GLU-D. And interesting, GLU-D, which is named GLU-D for this reason, is homologous to glutamate receptors, to AMPA and NMDA receptor. It looks exactly the same. And so we asked whether GLU-D might be the transducers. And we asked this by using a CRISPR approach, deleting GLU-D1, since GLU-D2 is not highly expressed in the subiculum. And this CRISPR approach is in such a manner that we basically take hippocampal cultures, we do acute deletions. So these are not mice, these are done acutely, and you can see that this is very effective. After about 2 to 3 weeks in culture, there's almost a complete loss of GLU-D1, and that allows us to analyze the effect. Again, there was no effect on synapse numbers as shown here, but when we measured synapse function, as you can see here, there was a tremendous decrease in NMDA receptor responses and an increase in AMPA receptor responses, exactly the same as the genetic deletion of the cerebellin 2, the cerebellin 2 deletion. So that leads us to the model whereby presynaptic norexin 1 and norexin 3 SS4 cause an upregulation of NMDA receptor and a downregulation of AMPA receptors via miraculously, magically, the same intermediate cerebellin 2 GLU-D1 complexes. How does this work? So the only way we could imagine this could possibly work is if GLU-D1 differentially transduces the presynaptic signals, which means it must have different sequences that are basically transducing this. And those sequences most likely are cytoplasmic, because that's what regulates, cytoplasmic factors regulate AMPA and NMDA receptors. So we tested whether distinct cytoplasmic GLU-D1 sequences might differentially control AMPA and NMDA receptors. We aligned different GLU-Ds from different species and found 5 conserved sequences. We made these mutations in these 5 sequences. the same mutations, and when we analyze them in rescue experiments. And as you can see here, in blue, there's the typical GLUD1 knockout phenotype, where you can see this increase in AMPA receptor responses with the GLUD1 knockout. All of the 5 mutants that we analyzed rescued this phenotype, except for one, mutation number 4. When we did the same analysis for NMDA receptors, you can see again that there's this classical decrease in NMDA receptor responses with the GLUD1 knockout. When we analyzed the mutants which rescued, there was only one that didn't rescue, mutant number 3, a different mutant. So these experiments demonstrate that different sequences in the GLUD1 molecule are responsible for mediating the signal transduction. And this leads us to this model whereby GLUD1, via distinct sequences, transduces the different Neurexin 1 SS4 plus and Neurexin 3 SS4 plus signals into either an AMPA or an NMDA receptor response, as illustrated here. GLUD1 is actually similar structurally to NMDA and AMPA receptors. It's the same family. They have the same structures, leading us to ask whether GLUD1 operates just like AMPA and NMDA receptor by a conformational change in its transmembrane regions. So we tested this by just taking away all the transmembrane regions and replacing them with another sequence, from another receptor, CD4, a single transmembrane region. And as you can see here, we did this for both GLUD1 and GLUD2 to test if they have the same functions. In these constructs, we are from GLUD1 only the N-terminal binding site for cerebellum and their cytoplasmic tail. And what you can see here is the same phenotype again as before with the AMPA and NMDA receptors upon the GLUD1 deletions. And in every case, the minimal construct that contains only the cerebellum binding site and the cytoplasmic tail of GLUD1 and or GLUD2 fully rescued. So the mechanism is totally different from AMPA and NMDA receptors. Only thing you need is cerebellum binding and the cytoplasmic tail. So then we were further interested if we could minimalize this even more, if we could generate a series of additional minimal constructs which would have only peptide sequences from the critical sequences of GLUDs. And I'm not going to go through this in detail here. These minimal constructs contain only the N-terminal cerebellum binding domain of GLUD1 and peptide sequences from the cytoplasmic tail of GLUD1 that include the 4 to 8 residue peptides that were mutated in the previous experiments. And what you can see here is that when we do this, for the AMPA receptor responses, again, there's this increase with the GLUD1 deletion. There's only one peptide that rescues it, which corresponds to the same motif whose mutation causes a decrease in the rescue in the full-length molecule. And for the NMDA receptor responses, same thing. Now it's a different peptide, but also. So quite amazingly, a very short cytoplasmic sequence coupled to a cerebellum binding domain's extracellular is sufficient to mediate this dramatic change in AMPA and NMDA receptor responses. And the way this works, we hypothesize, and we would like to test in further experiments, is that what happens is that cerebellum binding to neurexins causes a dimerization of GLUD that is differential between neurexin 1 and 3 because the affinities for these binding reactions are different, and that explains how this works. So I've tried to tell you in this first part, which was by far the longest part of my presentation, one particular function of what neurexins do. And let me tell you, this function is important because the regulation of NMDA or AMPA receptor responses at a synapse, at a central synapse, is key to synaptic computations. That determines not only the strength of a synapse, but also the relative contributions of electrical versus calcium signaling postsynaptically. So in terms of understanding how the brain processes information, regulating postsynaptic glutamate receptors is a key component. Let me now tell you about a completely different function. I will make this brief to show you a completely different way of how things can work. And this particular function also involves alternative splicing. I've shown you previously in my first part how glutamate receptors are regulated. Now I want to tell you about a function that relates to a presynaptic property, namely the release probability, and involves a different form of alternative splicing, namely alternative splicing at SS2. At many synapses, when you delete neurexin 3, you get a decrease in synaptic strength because the presynaptic neurotransmitter release probability is decreased. And we studied this in olfactory bulb synapses. These are olfactory bulb synapses in culture. You can see this large mitral and tufted cell that's being impaled by an electrode, and you can see a granule cells surrounding this. And when we analyzed neurexin 3 knockout neurons, as shown here with controls, these are conditional knockouts, we found that AMPA receptor responses are normal, NMDA receptor responses are normal, but that inhibitory responses mediated by granule cells are decreased by more than 50%. And this was the starting point of this particular project. So we wondered, how is the presynaptic release probability, that is the cause for this decrease in synaptic strength, decreased? And rescue experiments showed how this worked. We did many rescue experiments to whittle down what exactly the domains are of neurexin 3 that are essential. I'm not going to go through these experiments with you because of reasons of time. Instead, I will just show you two constructs that are the key constructs: the full-length neurexin 3 alpha on top, and what you see at the bottom is a minimal construct that contains neurexin 3 C-terminal sequences and a single LNS domain, the LNS2 domain. And what we analyzed are various splice variants in this minimal construct and tested whether they could rescue. And what you see here at the bottom is an input-output curve of the inhibitory synaptic responses, with the deletion in red, the wild-type delta Cre in black on top. So you can see this massive phenotype in synaptic strength upon the deletion of neurexin 3. When we rescue this with a minimal construct that contains an SS2-, it is fully rescued. This minimal construct is just as good as the wild type. However, if we have an insert in SS2, it is not. And this is also quantified by the coefficient of variation, which shows that this is due to a change in release probability. In red, the release coefficient of variation goes way up when you delete neurexin 3, and that means the release probability goes way down. You can rescue this with the SS2- in pink, but not in magenta with the SS2+. So here we have a phenotype where a presynaptic deletion of neurexin 3 causes a change in release probability that is rescued by a minimal construct in a splice-specific manner. When you have a release probability change, you would think that this would be a cell-autonomous phenotype, that a presynaptic molecule, when you delete it, causes a change in release probability. That's because the presynaptic molecule regulates release. But the fact that the rescue depends on an extracellular alternative splicing event suggests that it's not a cell-autonomous event, but that there must be some postsynaptic interaction that is necessary for the presynaptic release probability change. In other words, this result that SS2 alternative splicing controls the presynaptic release is puzzling and suggests an extracellular ligand interaction. Now, when you look at what actually SS2 regulates, it turns out it regulates dystroglycan binding. Dystroglycan is the only known neurexin ligand that is specific for SS2- and is inhibited by SS2+. And so we tested this. Some years ago, we showed that neurexins bind to dystroglycan in a manner that depends on SS2 minus, as shown here. And this was a black and white result, which led us to use CRISPR deletions again, acute CRISPR, just like I showed you for the GLUD. CRISPR is great. And what we found, as shown here actually in acute slices, not anymore in culture, is that the dystroglycan deletion phenocopies the neurexin 3 deletion. You get a decrease in synaptic strength in inhibitory synapses and an increase in the coefficient of variation. That means the decrease in release probability. And so these experiments that I've demonstrated to you here in a very abbreviated form show that you need the transsynaptic interaction with postsynaptic dystroglycan in order for neurexin 3 to regulate the presynaptic release probability. It's a transsynaptic interaction loop. It's again a transsynaptic regulation, transsynaptic signaling that determines the properties of these synapses. The same mechanism operates not only in the olfactory bulb, but also in the cerebral cortex, and most likely in other synapses as well. But it's specific to a subset of inhibitory synapses that actually express dystroglycan, which is not in every synapses. So, as a coda, let me just, to finish this up, tell you that what I've told you is obviously not the whole story. I've only shown you or focused on two particular interactions, two particular properties. Neurexins physically interact actually with another family of presynaptic hub molecules that are called PTPRs, or LAR-type protein tyrosine phosphatases, receptor protein tyrosine phosphatases. And this interaction, that is an interaction mediated probably by heparan sulfate modifications, suggests that possibly they may be related to each other. And indeed, when you look at synapses and the localization of these molecules, as you can see here in hippocampal synapses using DStorm, both neurexins and PTPRS, one of the LAR PTPRs, are localized again to nanoclusters, as I showed you before. And in these particular synapses, virtually all synapses contain either one or two or both of these nanoclusters. In fact, the majority of synapses, about 60%, contains both. So they're in the same place, suggesting that they might be redundant. And so we tested this by generating conditional sextuple knockout mice. In these mice, we are able to delete all neurexins and all LAR PTPRs using Cre recombinase. And we analyzed this in a cerebellar circuit. That is the synapse that is formed by Purkinje cells onto deep cerebellar nuclei, neurons, an inhibitory synapse, as shown here. And when we did this, as quantified at the bottom, we found that the individual deletions of either all neurexins or all LAR PTPRs did not cause a change in synapse numbers. But the double deletion caused a decrease of about 75% of synapse density, suggesting indeed that there is a dramatic redundancy, at least in terms of synapse numbers, between neurexins and LAR PTPRs. And this was confirmed using electrophysiology. What you see here are mini measurements. For reasons of time, I'm not going to go into that in detail. And at the bottom, you see the corresponding measurements of evoked iPSCs in the sextuple knockout. So this demonstrates that indeed there is redundancy. So in looking at how synapses are organized by transsynaptic signaling, not surprisingly, some functions of the many functions of neurexins and so on are specific to particular molecules. Other functions are probably shared. There are caveats here. Caveats are that some synapses remain. Again, this is not the whole story. It's not that these two families of molecules together account for all synapses. Individual knockouts often exhibit severe knockout phenotypes in synaptic transmission, even though there's no loss of synapses, suggesting that synapse loss could be secondary. So with these notes of caution, the only thing we can really conclude from these experiments is that there's redundancy. And I want to conclude here now with a sort of general idea, which I like to illustrate with this picture of a Swiss Army knife, namely that neoexons and LAR-PTPRs probably too, and many other molecules as well, are basically signaling platforms. They are platforms that are designed to do many different things in a regulated manner. Just like in a Swiss Army knife, where you can pull out different features depending on what you need. In the neoexons, there are different functions that depend on distinct ligand interactions, which often actually trigger these functions. So there's recruitment of calcium channels to the active zones. There's postsynaptic AMPA receptors I discussed with you. There's postsynaptic NMDA receptors. There's presynaptic receptors that I haven't discussed, and presynaptic BK channels that are regulated by neoexons. There is LTP, which is independent of NMDA receptors, which is highly dependent on neoexon 3, at least in the hippocampus. There are GABA receptors. And again, I haven't discussed this. There's actually a very strict regulation of the endocannabinoid system, which is quite fascinating. And all of these things depend on specific ligand interactions that are differentially regulated by alternative splicing, causing this network of protein interactions that is instrumental in dictating the properties of synapses, in teaching synapses what kinds of properties to have, and instructing synapses, instructing synapse properties. And we believe that this is a key feature of how the computational properties of synapses are being shaped. And I would like to end my discussion with you today by acknowledging just on time, despite the interruption, the major contributors to the work I discussed. Jinya Dai, who is now an assistant professor at Mount Sinai in New York City, performed most of the experiments on the SS4 alternative splicing and AMPA and NMDA receptors together with Jason Aoto, who initiated the project and who is now an associate professor at the University of Colorado. Justin Trotter did the super resolution microscopy. Alessandro Sklip did the heroic experiments with the LAR-PTPRs. Cosmos, Mann, and Lulu made major contributions at this Pang for the analysis, especially of the neoexon 3 mutations. And we've had a wonderful collaboration with a number of laboratories, in particular Xiaowei Zhuang at Harvard. Happy to take any questions and hope I could communicate to you, at least in a very abbreviated form, why this one family of presynaptic adhesion molecules is central to shaping the synaptic computational properties. Thank you very much for listening. Happy to answer any questions that there might be.
Thank you so much, Professor Südhof, for your fascinating talk and informative talk that introduced how the synapses are formed. So now is the Q&A section. May I invite our Director of MHRC, Professor Hector Zhang, to chair the Q&A section. So for all audience, you can raise up your hand and then we can unmute you and then to ask the question, or you can type your question in the Q&A box.
Okay, thank you, Sanatha. And also thank you, Professor Südhof, for this very interesting and I would say that highly cutting edge research on the the mechanism of neurexins and how it will be affecting, the transmission of our new signals in our neurons. So we have, 15 minutes of Q&A, and I will be asking the first question. And if you have any questions, and if it is, a non-technical part or, or more layman approach, you can just type in and I will be happy to ask for you. But if you are, doing similar kind of research, just like what Professor Südhof has presented, and you may request to raise your hand, and we will be trying to locate you, because we have more than 1,000 of participants in this online room. And okay, so I'll be asking the first question. So thank you, Professor Südhof again. Now I'm a practitioner and also more applied researchers. I would say that not many people may be doing similar kind of technical research as what you have presented. And most of us are more applied researchers and also practitioners in the field of psychiatry and also human services, that may be involving the brain. So my question is that is more layman, is that can you just try to summarize and give us some kind of take-home message, and how the mechanism of neurexins that you have presented will be having some kind of implications to our understanding of the brain, and also some kind of new psychiatric disorders, just like what you have talked about, for example, autism and also schizophrenia. So Professor Südhof.
Great question. As you, I'm sure, are aware, the last decade has witnessed tremendous progress in understanding the genetics of neuropsychiatric disorders. But it has left us with a conundrum. There's lots of genes, some of which are more frequently mutated than others, of which the neurexin is one of the most frequently mutated family. But we have no idea what these genetic changes actually mean. So for lay people, the geneticists often try to summarize this in Go analyses and all these diagrams, but the fact is that that's all humdrum. We don't really know what it means or why, because we don't know what the genes do. Okay, some of these genes are actually interactors of neurexins as well as neurexins. For example, neuroligands, AMPA receptors, and NMDA receptors, all this are part of that. So I think that for psychiatrists to have any hope of in future being able to actually treat disorders that are associated with mutations, what I was trying to explain is that we have all this genetics, but the genetics doesn't tell us about anything about the biology.
Correct.
There's no way we can translate the genetics to therapeutics. We need the biology. The examples I've shown you is that the biology, you can't just, unfortunately, a single gene doesn't have a single function. We need to figure out which ones of the functions, for example, of the neurexins, are actually the ones that drive the disease. Once we know that, we can try to identify therapeutic targets. So my take-home message would be, yes, the progress had been great. The genetics is fantastic, but it's a tiny step. And what we need to do next in this field to make progress towards therapies is to understand the pathogenetic pathways that are dependent on the genetic changes. And that will require actually doing deep biology, because there's no data mining or other approaches that will tell you. For example, if you do transcriptomics, which obviously we and everybody else has been doing, there's really nothing that comes out of that. You really need to actually look at signaling pathways, and you need to try to understand which circuits in the patient's brains are particularly vulnerable to these changes, and how you can potentially actually remedy this using pharmacological approaches. Does that make sense?
Yeah, sure. Yeah. Okay, so we still have time to take some questions. I still have not seen any message on the question box. Or do any of you have any question? You just raise your hand and we'll try to locate you and try to unmute you. So anybody would like to ask any questions to our speaker today?
Can I ask a question here?
Sure,
Because I
sure,
cannot
sure,
type,
Sonata.
yeah.
So go ahead, go ahead.
Yeah. Professor Südhof, so as you present that the neurexin play a key role in the synapse formation. So for follow-up your conversation with Professor Zhang, so can we target neurexin splicing variants in different psychiatric disorder as a treatment? That maybe a different mutation in neurexin might link to different neuropathological presentation in different patients.
Yeah, great idea. That would certainly be one avenue if we felt confident that that would actually help, for which we need more information.
Yeah.
But certainly there are now many disorders for which manipulations of alternative splicing looks like a terrific therapeutic avenue. You know, obviously started with SMA, where this has worked beautifully. And clearly that is certainly one avenue, one we could go, especially since, as Roche's drug has shown, you can even use small molecules to potentially manipulate alternative splicing, which is amazing. Yeah. And so I would see this as a wonderful outcome. In order to do this, however, we need to figure out which splice variant for which neurexin where. Most likely neurexin 1. What we don't know is whether you would, so the idea would be that you have a dysfunction of NMDA receptors, a decrease in NMDA receptor responses in subsets of circuits of the brain, those subsets in which neurexin 1 cerebellum GLUD1 pathway operates, which is only a subset. It's not all, okay? And then to specifically enhance NMDA receptors function in those subsets. And I think that that's probably the best we can think of right now in terms of a therapeutic avenue. Absolutely.
Yeah, sure. And we still do not see any questions typed in the question box. So I'm asking again, so any of you who would like to raise questions, please put up your hand and we try to locate you and unmute your mic, and so that you can ask directly your question. So anybody would like to ask any questions?
Nobody has any questions. It was so crystal clear.
No, I guess maybe, you know, highly or too technical and too cutting edge, and and that is why I try to. Oh yes, there's a question that I try to speak, and sorry I don't have a microphone, so that I have to type. Okay, very good. And I'm particularly interested in aging, as Professor Südhof concluded in the last few slides that neurexin can be considered as a Swiss Army knife, a platform for regulating synaptic responses. Can you briefly describe how would these mechanisms be affected by aging? So for your reply.
Yeah, I'm also very interested in aging, as Sonata knows very well. We believe that as we age, the mechanisms by which neurons communicate at synapses change. Primarily, probably because synapses are lost. I don't think that there is any indication at this point that there's a change in alternative splicing as a function of aging. There's been lots and lots of transcriptomics done on aging mice and even on aging humans. And most of the transcriptomics, unfortunately, doesn't allow you to tell whether there's a change in alternative splicing, because most of the transcriptomics is done as a very low sequencing depth. And so to look at alternative splicing by transcriptomics, you can't just use DropSeq or 10x genomics or something like this, but you have to really use very deep sequencing because otherwise you don't pick it up. And so I can't exclude at this point that there is a change in alternative splicing as a function of aging, but people haven't seen that as far as I know up to now. I think they may have missed it because of the limitations I just mentioned, but there is at least no evidence that it does exist.
All right, okay. So I've got another question. Is that can hormones from peripheral organs perform a similar function to neurexin?
Hormones from peripheral organs can influence the state of the brain tremendously. So what I didn't discuss is a really important issue in understanding how the brain works, which is the so-called action of the neuromodulators. So I talked to you about synaptic networks, but these synaptic networks are subject to regulation by neuromodulators. And these are not only peripheral hormones, but also locally released neuromodulators. You know, like, for example, serotonin or dopamine. These are classical neuromodulators that have massive effects on the behavioral state of an animal and a person. And they do affect these mechanisms dramatically. I would love to know if they actually regulate alternative splicing, which I don't know, because again, all the transcriptomics doesn't tell us because it's in the sequencing depth is too low. But I wouldn't be surprised if it did.
All right, okay. So I think we can still take one more question, if any. I do not see any questions typed on the question box. So I'm asking again, you know, any of you would like to raise questions, please raise your hand and try to locate you. No? Now, if not, and so I think I would like to thank Professor Südhof again on behalf of PolyU and also the Mental Health Research Center. And I'm wearing two hats, and I'm also head of the Department of Rehabilitation Sciences, and where we train occupational therapists and physiotherapists. And so I think what Professor Südhof has presented, I think, is an attempt to understand further the brain mechanism and how it works, especially the transmission of the signals in the neurons, especially the importance of neurexins and how it can be affecting the brain functions. I think it is a very important area. So I would like to you know, invite Professor Südhof to continue your collaboration with us. And for this time, this presentation is online. And I hope that in the future, and we can invite you face-to-face and your physical presence in Hong Kong and visit our university and visit our research center. And so that we can have further collaboration with you. And I hope that your research, very fundamental research, will one day translate to some kind of pharmaceutical or non-pharmaceutical kind of intervention, and so that, you know, people suffering from different kind of psychiatric disorders can benefit from your research. All right, okay. So Sonata, do you have any further conclusive remarks? You have to unmute. You have to unmute yourself.
Oh, yeah. Thank you so much, Professor Südhof, for giving us a fascinating talk again. So now I think we thank everyone for attending this Nobel Laureate lecture, and also we look forward to seeing you all in our coming MHRC events. Thank you so much.
Okay.
Have a good evening and good
Bye.
day.
Bye, Tom. Bye.
Thank you.
Thank you.